These first denizens of life were called prokaryotes, single-celled microbes that lacked a nucleus and had no organelles. They were primitive, but by no means simple. Prokaryotes possess all of the features that are required to fall under the definition of life. They were autonomous, completely self-sustained, and not dependent on other organisms. They were also metabolizing, or simply consuming chemicals and transforming them into energy for their bodies. Finally, prokaryotes were complex in their organization, able to evolve, develop, and most importantly, reproduce. Prokaryotes became living organisms when they obtained the mechanism that allowed them to replicate and pass on their genes. At first, such a mechanism was a single-stranded RNA molecule that was able to produce its own copies and become a self-sustained chemical system. RNA was able to store the information, or blueprint, of how to build a copy of an organism, as well as replicate itself according to this blueprint. The weak spot of RNA was that it was relatively unstable and easily damaged by the catalysts, or enzymes, necessary for metabolism. The solution was the emergence of a much more stable, double-stranded DNA molecule that was essentially a double RNA with a different sugar. DNA became the reliable storehouse of genetic information, while RNA became the workhorse that participated in the transcription and translation of the information into the actual production of proteins, as well as the replication process. These first microorganisms appeared and thrived in a world that would have been deadly for us or any other multicellular animal. They survived on the chemical nutrients in anoxic environments, and were likely resistant to the deadly ultraviolet radiation coming from the sun, with no ozone layer in the atmosphere to block it.